lesson in this section where we're going to just take a look at a couple of technologies that are related to DNA. One of the reasons we do de facto presentations is because we want to give you the opportunity to investigate other technologies that maybe we don't have time or that I don't have as part of my lessons. But the stuff I'm going to show you now could make good fodder for de facto presentations. So the first thing I want to talk to you about is genetic engineering, which I'm guessing you've heard of in some context before. It's not like it's a radical new thing. Maybe 20 years ago when I started teaching, it was kind of a cool, radical new thing. But this is a pretty commonplace thing these days. But there is still lots and lots of research and lots of new technology being developed in this domain. You could do a de facto about this. Now, essentially, here's what genetic engineering is. It's where we make recombinant DNA. Recombinant DNA is the result of the manipulation of genetic material. In most cases, we're going to take DNA from some organism and put it in some other organism's DNA. That's the end result. And so what I am going to show you is at the very base kind of how this process would work. Of course, there would be more details in real life, but in general, you need first something called a restriction enzyme. This is yet another enzyme in addition to the ones we saw for DNA replication and for transcription. So in DNA replication, we had like primase, helicase, DNA polymerase, ligase. Here's another enzyme. There are lots of different types of restriction enzymes. So this is not one single enzyme, it's like a category. These enzymes catalyze the cleavage, and that means cutting, separating, of DNA at precise nucleotide sequences. And I would need to use these enzymes in the donor, so the organism who is going to give a chunk of DNA, and in the host, the organism who's going to receive a piece of DNA. So if we look at my cutesy little picture on the side, the scary looking sharp teeth dudes that look like maybe evil Pac-Men, those are supposed to represent the restriction enzymes. They don't have feet or eyes or teeth in real life. This is just for the purposes of humor. But the restriction enzymes, as you can see, will come along and cut, where the arrows are, precise locations on a DNA double helix. There is a white and a black side of the diagram to represent that we're looking at two chunks of DNA. Let's just say, for saying's sake, that the left side, the white side, that's the host DNA, the organism who's receiving some DNA. And on the left or the right side, the black part, that's the donor. What we're trying to represent is that we need to cut the exact same sequence in the host and the donor's DNA. Why do we need to do that? Well, we want the piece of DNA that we're taking from the donor and giving to the host to fit perfectly. And so we need to cut the exact same sequence. Now, if we use these restriction enzymes properly, we'll get to picture 2, and we'll have what we call cohesive or sticky ends as our fragments. So when those restriction enzymes cut, notice how they're going to leave, both of them, some unpaired nucleotides at each end. And I know it's tiny, but on this end, the unpaired nucleotides are T, T, A, A. And on the other side, the unpaired nucleotides are A, A, T, T, the exact complement of the other side. So these fragments, these cohesive or sticky ends, they're unpaired, but they're the perfect complement to each other. So if I snip a piece of DNA from the donor and put it in a container where the DNA from the host has that same little sticky end, they will naturally match up and form hydrogen bonds. Now the last thing we need is ligase to glue them together. I didn't repeat what ligase is because it was part of the description for DNA replication, and it's doing the same thing. It's the gluing enzyme, the one that helps attach nucleotides to each other. Okay, so I didn't repeat its description because we already knew what ligase was. So this is the basic procedure. Use a restriction enzyme. It cuts a very specific sequence on the host and donor. Put them together and they will pair up because DNA nucleotides do not like to be unpaired, and they will form hydrogen bonds. Now, does anyone want to ask anything about this? This would be, for example, how we take the gene for insulin and put it into a bacterial cell so that we can produce mass quantities of insulin. This would be, for example, how scientists took this gene that fish have that allows them to not freeze in really cold water. They call it the antifreeze gene. So they would use restriction enzymes, snip that gene out of the fish, and put it in plants so that plants can be resistant to really cold weather. So those would be some cool examples of applications of this. Now, the other thing that I want to show you today is about a way that we can analyze DNA. So the first technology was manipulating the DNA to do something else. This is a way that we can analyze DNA to figure out from which person or which organism did a DNA sample come. The technology is called gel electrophoresis. If I was going to ask you about it, I would probably write the word gel electrophoresis and not be super concerned if you couldn't spell or remember the exact name of the process. Essentially, what we do in gel electrophoresis is we're trying to take pieces of DNA and separate them. So there's a little picture here that's trying to illustrate the steps. The first picture shows you a rectangle. What it really is like a plastic container with what looks like clear jello inside of it. So there's like this jelly gel fluid. There are some holes at one side that we call a well. And in each of those holes, we would take a sample of DNA and we would put it in the well. So you can see up here, I've got some little syringes or micropipettes, and I'm putting these blue samples in each of the wells. Now, where did I get these samples? Well, I'm trying to separate molecules according to their mass and charge. That's what gel electrophoresis does. And so what we do is we would take a sample of DNA and try to turn it into fragments. We would make these fragments with the same restriction enzymes I talked about on the last slide. They snip at particular sequences. So let's say you're a crime scene investigator. You could collect a sample of DNA, use some restriction enzymes to snip it up into little pieces, and then we could put it in one of the wells of our gel electrophoresis because we're going to try to figure out who the criminal is. So we take those fragments, we place them at one end of the gel, and then I don't know if you can see, it's tiny, there's like pluses and minuses here. That's to indicate that we're running an electrical current. So we would have to plug it into some power, turn on the current, and when the current passes through the gel, it attracts DNA fragments to move to the other side of the gel. The smallest fragments move faster, so really small fragments will move really far, so they'll get maybe almost all the way to the other side of the gel. The idea is that the banding pattern that we get, based on how far each fragment moves through the gel, gives us a genetic fingerprint. So on the right, that's a real picture of an actual gel electrophoresis that was run. And the way we would use it is we would try to match up one banding pattern with another, kind of like what we did for chromosomes, which chromosomes were homologous when we looked at their length and banding pattern. So for example, on this gel, the one on the left, that's usually where you place your like unknown sample, the one that you're researching. So we could say the one on the left, that was our crime scene. And we're trying to match it up with one of the other banding patterns to see who the criminal or maybe the victim was at this crime scene. And if I look at each column in the gel, I might see some bands that line up, but I would see a lot that didn't. Until I got to the very last one. The very last one has the same banding pattern. So if the first one was my crime scene and the rest of them were all potential suspects, I would say whoever the suspect was in that last column, that's my guy or that's my gal. That's whose DNA has the same banding pattern as the sample that I had. Now, would anyone like to ask anything here about gel electrophoresis?